paper chromatography. Paper chromatography again can be done in 2 dimensions, single or 2, and the force that is used here is called as capillary force. So you have an apparatus where you just take a small beaker, or you have an electrophoresis chamber, where again in this you will have a solvent system. The solvent system is the mobile phase, and then the sample is moved, that is your solute. So the mobile phase differs depending upon what you are separating. So you will have a different mobile phase for amino acids, different mobile phase for carbohydrates, different mobile phase for bases, nitrogenous bases. You select a mobile phase in which your compound can get separated. Basically, the kind of paper we take is called as a Whatman filter paper. A Whatman filter paper is taken, you put your sample and allow it to run using a capillary force. Now, the mobility of the compound is calculated with what is called as a retardation factor, that is, which depends upon the molecular weight of the compound, that is called as an RF value. So, you calculate RF value using the formula distance travelled by solute by distance travelled by solvent. Now, this is like a fingerprint. Now, every compound will have a different RF value. For example, you're talking about amino acids. Each amino acid will have a different RF value. But understand that paper chromatography is a very laborious, time-taking process, because if you're taking a big sheet of paper and allow your amino acids to run, they might take about 6 to 8 hours for an amino acid to run on the paper. So, it's a kind of very tedious process. And now with the kind of sophistication in chromatographic techniques available, we are no more using paper chromatography, but we have moved on to the more sophisticated column chromatographic techniques, in which we also have kind of a thin layer chromatography. Like even thin layer chromatography can be considered as a part of your partition chromatography. In thin layer chromatography, you will have a glass plate on which you have a silica coating. On this, you load the sample and allow it to move. Yeah, same thing, you can calculate the RF value. Now, the major difference between a paper chromatography and a thin layer chromatography, in paper chromatography, the water that is present in the crevices of the paper will act as a solvent system. Now, because water is present, suppose you are trying to separate non-polar substances, more specifically if we talk about your lipids, which are non-polar substances, they cannot be separated on a paper chromatogram. That is why you will take a more organic compound which can help the separation of both polar and non-polar substances. So silica is a kind of organic substance which helps in separating both polar and non-polar. So on a TLC you can separate amino acids and also lipids. So that is the benefit when you compare TLC to paper chromatography. Then you're talking about gel filtration chromatography, very important one. This is also called as a size exclusion chromatography. This is a type of your column chromatography. So column chromatography, you have a column in which you will pack this. You will have commercially available beads that are available based on size. Okay, so like for example, you can calculate, suppose you want to, Cefidex is a commercially available gel filtration. So suppose you want to take Cefidex G250, that means the exclusion limit, the molecular weight of the compounds that you can separate, anything that is less than 250 Daltons, you will get separate. Now, how does a gel filtration chromatography work? This is exactly like your molecular sieve. So, that is when you take a sieve is kind of your jallida that you take, or a channi. You put anything. Now, when you are using the sieve, what happens when you use the sieve? Anything that are larger than the size of your sieve mesh, or smaller than the size of the sieve mesh, they all will retain on the top of the mesh, and everything that is larger than that will fall down. Okay. So now what you doing? You're taking a cutoff limit. Suppose you are sieving very fine granules, you will take a mesh which has a very small pore size. So all that you require either goes down or is retained on the top of the mesh. Unwanted things are retained, and what you need, the fresh atta, comes down of that particular mesh. That's exactly what is done in gel filtration chromatography. You choose a matrix which has an exclusion limit of your compounds. So suppose, let's say you are separating a protein solution, and then the protein that you need has a lesser molecular weight, and all the other contaminants have larger molecular weight. So you select a sieve size where, when you fill that particular column with your matrix, all the compounds which are larger will pass through the gaps that are present in the matrix and will come out first, but your protein which is lesser than the cutoff limit of the bead size will enter into the beads, slowly get separated and come out in the last. So whatever comes out last is your pure protein. All other contaminants would have been separated in the initial stages. That is a simple principle that we use in gel filtration chromatography.